आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़।
We bring you all a podcast series on STEM popularization, that is, science, technology, engineering, mathematics, and medicine, supported and catalyzed by the National Council for Science and Technology Communication, the Government of India. Program is produced by Media for Community Foundation.
This is Manjeet. Welcome back to the Cefipra podcast. In this episode, we continue exploring groundbreaking collaborations between Indian and French scientists. Now let's listen to our great scientists for more information.
Hello viewers, I am Minhaj Sirajuddin. I am a faculty at INSTEM, Bengaluru. My research interests are cytoskeleton and contractile machinery across scale dimensions, with a particular focus on heart diseases and neurological diseases. Our project, together with Professor Carsten Janke from Curie Institute Orsay, the project is called Control of Microtubule Dynamics by Tubulin Code. Now this is a mouthful. I will break it down into simple parts. So we wanted to understand how an important element inside our cells are being regulated. That is the crux of the project title. And if you imagine cell, which is the smallest unit of our human body, there are proteins which actually forms like a network or roads, railway roads, and you can imagine as a city as a cell, and there are railways and roadways, and there where you have trucks and cars and buses and motor vehicles moving in roads, and trains are moving in one direction, and that is what we call as cytoskeleton filaments. It provides a framework for the cell. At the same time, it also provides a framework for transport of molecules across small parts of the cells. I mean, you might think that, like, okay, cell is very small, but the cargo is even smaller, at least 1,000 times smaller. So it needs to go from A to B. So these are the filaments which we call it as cytoskeleton, and microtubule is one of them, which is an important one. Microtubule is involved in, for example, cell division, transport of many of the molecules inside the cell, and many other important functions they perform. And microtubules are also very important target for cancer therapy. There are many drugs which target microtubule cytoskeleton. For example, colchicine, Taxol, and vincristine are some of the famous drugs. Now, if you imagine in humans there are so many different types of cells, and each cell performs a different task, and this cytoskeleton also performs a different task in each different cell. So we have to understand how these are performing in different tasks. So for that, like, we generated a marker, and that was one of the highlight of our work. And this was possible only by collaborating with our French scientists, who are actually a world leader in understanding microtubule cytoskeleton polymer. And we teamed up with them because we had complementary skill sets. Our lab was more interested in biochemistry, structure, and other in vitro related work, and they are more expertise in cell biology, imaging, and also mouse models to study these molecules. So we teamed up, and then we had a lot of exchange. For example, the Cefipra funding actually helped us to collaborate, not just collaborate, but also have exchange of people from France to India, and at the same time materials were exchanged between our labs. We were able to send trainees across different labs so that like they can train and learn from each other labs and perform experiments. For example, in one case, we sent a lab member to their lab to perform a neuronal experiment, which was not possible for us here. At the same time, they were able to send a trainee, a PhD student, to our lab to perform a structural biology cryo-electron microscopy. In our institution, we had the cryo-electron microscopy facility, so we were able to get the high resolution structures, which was also published. So altogether, it was a mutual, wonderful collaboration. The CEFIPRA funded project resulted in several publications which are high impact, at the same time garnered much visibility. More importantly, we were able to identify a marker which is very well recognized across the world now, and it is available as a resource for other researchers to take up and study in their lab. And we have also patented this molecule for commercial interest. And in future, our collaborations have already set up pathways for exchange in other means. So thank you, CEFIPRA, for funding this project and making a fruitful research collaboration.
This episode is brought to you with the content support from CEFIPRA and the technical support from the Sparrows team. We extend our heartfelt thanks to the Director of CEFIPRA, Professor Nitin Seth and Dr. Payal, for their continuous guidance and support. Stay tuned for more episodes and thank you for joining.
आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ।